them seriously. 6 minutes. Your time begins now. Okay, thanks for joining us, Deasia. Hopefully that was enough time for folks to knock out the warm-up. If not, as with all of our assignments, you've got until Wednesday, February 10th, which is this coming Wednesday, to get them done and submitted. I really, really, really want to emphasize the importance of these warm-ups and exit tickets, because even though they aren't worth many points, they do give you a big bang for your buck. They give you an opportunity to see exactly what the test questions will look like. We've only got 2 standards in this unit. And so the test is not going to add all that much more material, but it can be confusing material. So you want to expose yourself as much as possible to these test questions. That's really what a strong student would do to prepare themselves to perform at their best and to master these tests. So good job to those of you who didn't, to those of you who did complete the warm up. To those of you who did not, please do make sure that you go back, make a mental note, write it down in your phone or in your calendar or on your notebook. Give yourself an opportunity to succeed. Today's lesson is entitled Meiosis and Genetic Diversity. Today is February 5th, 2021. It is the 4th day of our 3rd unit. The objective of today's lesson is to explain the role of meiosis in sexual reproduction and genetic variation. That is the same objective we had yesterday. Like I said, we only have 2 standards in this unit. That is one of them, to explain the role of meiosis and sexual reproduction and genetic variation. Yesterday, we really just walked through the steps of meiosis. We talked about the fact that it follows those same 4 steps of mitosis: prophase, metaphase, anaphase, and telophase, but it does those 4 steps twice. Today, we're going to add in a little bit more about the genetic variation piece. So I want you all to understand various sources of genetic variation. The essential question that will serve as our guiding light as we move through this lesson: how does meiosis ensure genetic diversity in sexually reproducing organisms? So I've always thought it would be a good idea to write down these essential questions. It gives you an opportunity to answer an actual question at the end of the lesson. If you can do it, great. You took away what you needed to take away from that lesson. If you can't, well, you probably have a little bit more work to do. You might need to reach out to me and say, hey, Mr. Red, I didn't quite get that. Can we spend a little bit more time going over this one-on-one? Or I had to step away from my Chromebook for a minute to use the bathroom, or to pick up my sibling who just fell on the floor, or to grab a snack. So I missed this part of the presentation. Can you go over it with me? Of course I can, but I need you guys to reach out to me to let me know what's going on. So these essential questions give you an opportunity to answer that question, to answer a question, to understand if you know something well enough or not. We're going to start off today, as we started off yesterday, with a quiz. This quiz is only 13 questions long. We should be able to move through it pretty quickly. I will share this screen. I will also send a link to the chat. But the game code is 050313. Or you can click on that link right there, or go to joinmyquiz.com and enter the game code 050313. 2 people in. Thanks, John and Lance. 3 people. Thank you, Araceli. Thank you, Janelle and Melissa. Thanks, JT. Thanks, Josue. We've got 7, 8 people. Thanks, Abigail. But we've got 17 in the chat, so we need 9 more. Now we've got 9. Thanks, Lisa. We need 8 more people. Where are we, folks? Where are we? We've got 9, but we should have a lot more. Sienna, where are you? Deasia, where are you? Abina? Janelle? There's Janelle. Sorry, Janelle. You're already here. There's Ashley. Thanks, Ashley. 7 more people. I'm going to start in about 10 seconds. Tayshawn, if you can join us, that'd be great. Thanks, Sabina. All right, so I'm going to go ahead and get started. If you were not able to join yet, that's no big deal. Of course, you can join as the game proceeds. Remember, yesterday I wanted us at an accuracy of 70%, and we achieved that goal. Let's see if we can get to an accuracy of even higher than 70% today. Ooh, we started off kind of rough. Araceli out to the early lead. Janelle and Abigail right behind her, tied at 880. Get that accuracy up, folks. Good job, Janelle. Okay, let's see if we can get that accuracy up a little bit higher, folks. I'd love to see it up at least above a 70%, which is where we were yesterday. I know we threw in this new concept of meiosis. One of those questions is probably difficult to see and therefore difficult to answer. So I apologize about that. But let's keep pushing upwards. We're at 68. Let's keep going. Josue is in the lead. Okay, we've got 9 out of 14 people done. Our accuracy is at a cool 75%. That's excellent, folks. Let's keep going as we wrap up our top 5. Looks like it's basically solidified. Someone might come in and try to jump over Janelle. Who knows? Janelle, you did quite well. Lisa surpassed you. Looks like probably because she was answering quickly. JT, excellent job. Abigail, excellent job. Looks like you maybe only missed one question. So what we'll do, I'll let the people who are still working try to finish up, and let's keep that accuracy up high, folks. I'll let those of you who are still working finish up, but those of you who are done, like I said, 9 of you are done. So we're going to jump back into the notes. We do have a good amount to cover today. So I want to make sure that we have adequate time to do that. So let me jump over into the notes again. All right, so what we're seeing here are fraternal versus identical twins. What's the difference between the two? Does anybody know? Does anybody know any differences between fraternal and identical twins?
Fraternal were born on the same day but look different. Identical, they look the same.
Yeah, so there's a clear physical difference in terms of how they look. Identical twins are called identical because they look identical, but the difference really lies in the genetics as well. So we'll talk more about this later in the course, but identical twins are not only the product of the same birth, but they're the product of the same egg. So they come from 2 different sperm cells, but those 2 different sperm cells fertilize the same egg. And so they have very, very, very similar genetics. Identical twins are the product of the same birth, but they came from their own sperm and their own egg. It just so happened to be that the woman, the mother, released 2 eggs at the same time. And so both eggs were fertilized, leading to fraternal twins. But fraternal twins genetically are no more similar than other siblings who may have been born years apart from one another. It just so happens to be that they're the product of the same birth. That fact gives them some additional similarities because they were exposed to the same environment for the first 9 months of their development, but it does not necessarily mean that they are genetically identical. It just means that they had the same birth canal, the same womb. So it's a difference. And if we can look at these images on the screen, we see identical twins on the right. Same egg, different sperm cells, same egg. Fraternal twins on the left. Okay, so clear differences, but these 2 girls on the left look very, very different. And you might even question how they have the same parents. So we're going to watch a quick video about this. And I want you guys to think more so about their differences. What causes their differences as we watch this video? Oops.
These pretty teens are best friends, and they're also something else. Something that may surprise you.
We're twins.
Believe it or not, they're twins.
Yep, we're twins.
We're
Same
twin
mum,
sisters.
same dad.
You heard right. They have the same mom and dad. What are the odds that one would be black and one white?
A lot of people are just in shock.
Lucy and Maria Aylmer live in Gloucester, England. Maria has a caramel complexion, brown eyes, and thick curly black hair. Lucy has pale white skin, ginger red hair, blue eyes, and freckles. Most people have the same reaction when they hear they are twins.
They say, how did it happen? We don't believe you.
So, how did it happen? Their mom, Donna, is half black and half white. The twins' dad, Vince, is white. The couple was thrilled when a sonogram revealed they were expecting twins.
I just cried. It was shock.
Mom did a double take when she delivered one black and one white baby.
I'm so glad Mum dressed us in such cute little outfits.
Mom dressed her adorable twin daughters in matching outfits, just like twins everywhere. But by age 10, Lucy says she didn't feel like a twin, so why dress like one?
We don't look alike, so why should we have to wear the exact same thing?
Maria says she once wished for Lucy's straight hair.
I used to cry about it. I hate my curly hair.
And Lucy says she was sometimes taunted at school.
They thought I was adopted. You calling me a ghost.
Now 18, the twins not only look different, they have very different personalities.
We're completely different.
Maria loves getting dressed up in chic clothing. She is very outgoing.
No, but I love meeting people. Like, I'm not scared to approach people or anything, so.
Whereas I'm terrified of like going up to random strangers.
Lucy prefers casual clothes, but they've come to embrace their uniqueness as black and white twins.
If I have kids one day, they might come out looking like Lucy.
Okay. So what are your thoughts, folks? Anything interesting about that video? It makes sense. Why does it make sense, Janelle?
The one thing that shocked me is how, like, the mother is half black and half white, and then like mostly one of the father's and the mother's genes they went to one child, and then most of the black genes kind of went to the black daughter, if that kind of makes sense.
Yeah, that makes sense. And I think that is one of the things that I was hoping you all would pick up on, is kind of this idea of race and how that came up in this video, because clearly one daughter is probably likely to be perceived by most people as black, and the other daughter is likely to be perceived by most people as white, but they have the same parents. And their parents' race is also a little bit more obvious. Obviously, the dad is probably likely to be perceived as white, and the mom, even though she is biracial, is also likely to be perceived as black. So we can see that the impact of how those genes came together resulted in two totally different daughters, and how the perception of them in the world is likely to be different. I also thought it was interesting, and I hope that you guys picked up on this, that they have different personalities as well. So I think too often we discuss genes as having implications on our physical characteristics, but there are also vast implications on our emotional, mental, and personality characteristics as well. But one thing that I have to state, and one thing that we'll talk a lot more about, is the interplay between the environment and the genes. This is always an ongoing and difficult question that scientists have to ask themselves. Which one of those two things, the genes or the environment, is having a greater impact on a person's outcome? It's almost a question that's impossible to answer. In fact, for most things, it is impossible to answer, because the two interplay. Your genes impact the way that you interact with the environment, and your environment certainly impacts your genes. We find that people end up with mutations as a result of the things that they are exposed to. The way that you're raised, especially in the first 10 or so years of life, has a huge impact on your outcomes, your life outcomes. People who are exposed to trauma at an earlier age, their brain literally develops in different ways. People who are exposed to many different languages at an earlier age, their brain develops in really specific and fascinating ways. People who are exposed to music at an early age their brain develops in fascinating ways. So your environment and the things that you're exposed to, not only in terms of the chemicals you're exposed to, but also in terms of the cultural norms and practices that you're exposed to, has a huge impact on the person you end up becoming, as well as your diet. Your diet is a part of the environment. It's part of what you're exposed to, but it also has an impact on who you become. So these are all things that I think we have to acknowledge. It's not just genes that determine who we are. Yes, your genes play a huge part in who you are, and some of who we become is predetermined from the moment that the sperm meets the egg. because those genes are carried already. But the environment is also going to play a large role. So in the case of these twins, I think that the environment plays a large role in who they became as well. One of them we heard say she's uncomfortable. She's horrified by the idea of approaching people. The other one talked about being really outgoing. So certainly those are qualities that are impacted by their genes, and they determine their personality, but also it's an impact of their environment as well. Perhaps they had some interaction in their earlier years in which one daughter was feeling a little more shy and the other daughter felt a little more celebrated and therefore more outgoing. So it's just a question that we always have to ask ourselves. How are the genes and the environment interacting with one another? Okay, so here we've got a Venn diagram, and I want us to try to fill out this Venn diagram. I love visual representations, as you all know. I love graphic organizers. I think they are an excellent way of trying to memorize things, and this unit in particular is heavy on the memorization. So I'm going to leave this up for about 30 seconds, and I'm not going to say anything, and I want you all to take out your notebook if you don't already have it out, copy down this Venn diagram, and then we're going to start to fill it in. Okay. So we're going to throw up some terms at the bottom of the screen here, and I want you all to tell me where that term goes. Does it go on the left with mitosis, in the middle, meaning it applies to both mitosis and meiosis, or does it go on the right, meaning it applies only to meiosis? So the first term, we've got two daughter cells. Which one of these processes produces two daughter cells? And you can just give me the first three letters. Good. Thank you, Abigail. Two daughter cells. Mitosis produces two daughter cells. So we're going to put that on the left. The next one, which one has two rounds of cell division? Good. Thank you, Janelle. You're quick with it. Yep, that's a meiosis process, two rounds of cell division. Which one is a type of reproduction? Good. That was kind of a trick question. I see some of you are jumping in now. A lot of you are jumping in now saying both. Good. Both are types of reproduction. Which one produces diploid or diploid cells? Excellent. That is a mitotic feature. Produces diploid cells. What is a diploid cell? How would we define that? Or what is a characteristic of diploid cells? Does anyone remember? What are diploid cells? So yeah, so mitosis produces two identical cells, but a diploid cell does not. I know what you're thinking about, Michelle, because you see that prefix di, meaning two. But a diploid cell does not necessarily have to have an identical. Okay, so make sure you guys write this down. Maybe I skipped over it earlier in the week. Diploid cells have two, remember di equals two, complete sets of chromosomes. Yes, thank you, Abigail. You kind of came in cleaned it up there. Yeah, they have paired chromosomes, 2 complete sets, one from your mother, one from your father. Two complete sets of chromosomes. All right, the next term. Which one produces somatic cells? Good. Thank you, Abigail. Yeah. And what might be an example of a somatic cell? Give me one example. Skin cells. Yeah. Thanks, Janelle. Can we think of any others? Blood cells, bone cells. Yeah. Everything that is not a sex cell is a somatic cell. So which one produces sperm and egg cells in that case? Wonderful. That would be meiosis. Which one is needed for sexual reproduction? Good. Everybody's jumping in now. Meiosis, meiosis. Wonderful. Yeah. So again, meiosis itself is not sexual reproduction. Meiosis is just producing the cells that make sexual reproduction possible. It produces sperm and egg cells. But by itself, meiosis is not the process of fertilization. Which one is needed for asexual reproduction? Yep, mitosis. That was kind of a gimme following. Which one undergoes one round of cell division? Mitosis, good. Which one produces haploid cells? Now, thank you, Janelle. Thank you, Abigail. Thank you, Melissa. Okay, so if a diploid cell has 2 sets of chromosomes, one from each parent, then what is a haploid cell? And to make it easier, you can give me an example with numbers. It has half of the full set. So in the case of human beings, our somatic cells, like the skin cells, the bone cells, those cells have 46 chromosomes. They have our entire genome. They've got chromosomes that we got from our mom and chromosomes that we got from our dad. Our our gametes, our sex cells, they have half of the full set. So they only have 23 chromosomes instead of 46. Good. Just wanted to clear that up. Which one produces unique daughter cells? And I see that word unique. Janelle just sent it. Good. That's meiosis, which means that the identical daughter cells obviously are coming from mitosis. So I want to make sure that you guys have this Venn diagram. It looks like at least 5 of you who contributed to this activity have a good grasp on the differences between mitosis and meiosis. But I want to make sure that everybody has an opportunity to get this Venn diagram down. So please take a screenshot, take a picture, finish writing it down. I'm going to leave it up for about 30 seconds to give you all an opportunity to capture this in some way. Okay. So our conversations today will center around meiosis and genetic variation. So what is genetic variation? Genetic variation is the diversity of gene frequencies between individuals or between populations caused by several sources. Remember, the words that are highlighted in yellow are the words that I suggest you write down at the very least. Today, we're going to discuss 5 of those sources that contribute to genetic variation. Now, it's important to also note, even though it's not highlighted, that it does say genetic variation can refer to individuals or populations. In this first part, as we first introduce genetic variation, we're going to think about it through the lens of individual variation. As we move through the course and we start talking about evolution, we'll think about it through the lens of population variation. So this image on the right of these, I don't know what they look like, some type of Spaniard or Spaniel. I'm not sure. Some of you might know dogs better than I do, but what we're seeing here is individual genetic variation. These 4 dogs, the little ones, the puppies on the right and on the left, are from the same litter, but we can see differences in them. Some of them have eyes that are bigger than others. Some of them have droopier eyes and droopier ears. Some of them have lighter coats than others. Some of them have straighter hair than others. And they both, clearly, all of them look different than their parents, which are the dogs that are sitting up a little bit higher. So what we're looking at here is individual genetic variation. These are individuals who are genetically similar, but they've got differences because of this process of meiosis and sexual reproduction. All right. So number 1, mutations. Mutations are the primary source of genetic variation. So that's why I wanted to mention them first. A mutation, I'm sure, is something you've heard a lot about, but I'm going to provide the scientific definition before we start talking about some of the sci-fi things. A mutation is a change in the structure of a gene caused by the deletion, insertion, or substitution of a nucleotide base, A, T, C, or G. Hopefully you can think back to not even a month ago when we were in our first unit and we introduced nucleic acids. I told you that nucleic acids are made of nucleotides, and nucleotides are made of 3 things. There's a 5-carbon sugar called pentose. There's a phosphorus group, and then there's also a nitrogen base. These bases are either A, T, C, or G. Those are the letters that we use to identify them. Now, in sci-fi, we think about mutations as giving people superpowers or, totally changing the way that they look. And, they come from these exotic sources, spider bites and vats of acid and things like this. But mutations are really frequent and they're normal. Our cells are imperfect, so they often get mutated through these processes of mitosis and meiosis. The thing about mutations is that they can be inherited, meaning that they can be passed on from one generation to the next. So the sources of mutation are not always exotic. Oftentimes, they are just the fact that you got it from your parent or you got it from being exposed to UV light from the sun, or you got it from your diet. And then once you acquire a mutation, it could potentially be passed on to your offspring. And I say could because not all mutations actually get passed on, and also not all mutations actually have any effect at all. So in sci-fi film and comic books and fan fiction, stuff like that, we hear about mutations that are really, really detrimental. They really hurt people. They make them really ugly. And then we also hear about mutations that are pretty cool. They give people these unique powers, superhuman strength, or the ability to bend things with their mind, move things. What's that called? Telekinesis. But in reality, most of the mutations that happen in your cells all the time have no effect at all, because the cell will just go in and correct that mutation, or the cell will just say, "Oops, I'm messed up. I'm going to go ahead and die." So another cell can come in that's healthier. Some mutations are beneficial. The classic example of a beneficial mutation, one that happened in a population over many years, it didn't just happen it didn't happen from one generation to the next. It happened as the population evolved over hundreds of thousands of years. But giraffes, evolutionarily, giraffes began as essentially tall horses, but their necks were no longer than the average horse's neck. Over time, because of environmental pressures, as there was more and more competition for food sources on the ground, giraffes that had longer necks got what we call selected for. So the giraffes with the longer necks had an advantage because they could reach food that was dangling down from trees rather than having to bend over and get food off of the ground, where other animals were also eating. And so over time, as that gene was passed on, the gene for long necks was passed on, the necks got longer and longer and longer, because the longer your neck, the greater the advantage. And eventually we get to a point where giraffes have necks that put their mouths 20 feet in the air. And it gives them an advantage. It gives them, yes, and this is a process called natural selection, Josue. I'm glad that you know that. It's something we'll talk a lot more about towards the end of the course. So that's an example of a positive, a beneficial mutation. And then there, of course, are detrimental mutations. Often these detrimental mutations only affect individuals, because if a mutation is so detrimental that it prevents a person from surviving, then of course they won't be able to pass it on, because they won't be able to have children. Or if it makes it less likely that they will have children, or if it makes their children less likely to survive, then that it's going to be difficult for that gene to be passed on. And then there are some that are confusing. They do both good and bad things. The mutation that causes sickle cell anemia also provides an immunity to malaria. So we've got a bad thing. Sickle cell anemia it can be really painful. It can be a deadly disease that affects your red blood cells. But at the same time, it gives you an immunity to malaria. The reason this happened is because, and the reason why it was continued to be passed on is because of that good reason. So people who have ancestors from Western Africa, those ancestors were obviously exposed to malaria because of the dense mosquito population passing on this disease. So those individuals who had sickle cell anemia and had immunity, they were able to survive. They couldn't get sick from malaria, so they survived. They had more kids. They passed on this trait. So that's a weird one. That's both good and bad, and the good kind of outweighs the bad, meaning that it gets passed on. But like I said, most mutations are ineffectual. We're going to talk more about these different types of mutations, so I don't ask you all to write down anything on these next few slides, but I do want to run through a couple of different types of mutations. So a simple type is called a point mutation. This is when one of those nucleotide bases is changed. It's kind of swapped out. So as we can see here, we've got a C where there really should be a T. Everything else is the same, as you can see. So there's only one nucleotide base that is messed up. Now, typically, these are the most often ineffectual mutations because the cell can pretty easily go in and fix this, or the cell can, like I said, just decide that it's not worth it, and so I'm no longer needed. I'll just allow for another cell to come in and take my place. So these mutations usually don't have a big effect, if any effect at all. Now we have to keep in mind that genes, that these DNA, the DNA is coding for a protein. The DNA is the instruction manual to produce a protein. So it's the protein that we really want to think about as being either messed up or still being able to function normally. Another type of mutation is called a substitution mutation. This is when a group of 3 bases is changed, and a group of 3 bases is called a codon. So as we can see here, there is a GAT where there should be a TGC. These mutations usually have an effect on the protein that is made. So swapping out a codon is typically not a good thing, and then oftentimes swapping out a codon is going to affect the protein that you can make. Each codon codes for a specific amino acid. So each group of 3 letters codes for a specific amino acid. And those amino acids, of course, eventually get chained together to make a protein. So if you change one of those amino acids, it's possible that you could change the protein. It's not guaranteed, but it's possible. The reason for this is that each of the amino acids has different chemical characteristics. Some of them like water, some of them don't. Some of them are positively charged, some of them are negatively charged. Some of them have no charges at all. So if you, let's say you put an amino acid that doesn't like water in a place where there should be an amino acid that does like water, that's a big problem. Now the protein is probably not going to function. So swapping out an entire codon is dangerous and often leads to some type of effect. Like I said, it could be a good effect. Maybe this actually helps the cell to survive. Maybe this actually produces a protein that helps the organism to survive. But it could also be a bad thing. And then one more type of mutation is called a deletion. So those codons that I was just talking about, those bases of 3, we read each of them in groups of 3 to produce an amino acid. So here in this example of the original sequence on top, we've got TAA. That is a codon. Then we've got CTG. That's another codon. We're reading them in groups of 3. Then we've got CAG. That's the third codon. But as you can see, we deleted a T. So when you delete that T, we still have to read in groups of 3. Starting from the same place, we still have to read in groups of 3. So now everything that came after that T is messed up. This first codon is good. TAA, that's the same codon that we had in the original sequence because it came before the deletion. And then we've got C, which is in the right place, but then we deleted the T that was supposed to be there. So everything is now messed up after that. Everything that comes after the mutation is altered. So instead of CTG, we now have CGC. That's messed up. It's incorrect. Instead of CAG, we now have AGG. So you can see how damaging deleting a single base is. And we can also think about it as inserting a base. That would be just as bad because we're shifting over our reading frame. We have to read in groups of 3. If we shift it over, then everything that comes after that shift is messed up. Okay. We'll talk more about that and do some practice problems later in the course. That's actually one of my favorite topics. And if that interests you, genetics is probably something that is one of those fields of biology you might be drawn to. A second source of genetic variation is called crossing over. And for my hooping fans, no, we're not talking about a crossover. It is different. Crossing over in terms of genetics is the exchange of genes between homologous chromosomes, resulting in a combination of parental traits. This only happens during prophase 1 of meiosis. We talked about this very briefly a couple of days ago. Crossing over does not happen at all during mitosis because, of course, we don't want genetic variation in mitosis. We want to produce copies. It would be bad if my muscle cells were going through mitosis and then they went through some type of genetic variation process and became skeletal cells. That would not be a good thing. So crossing over is only happening during meiosis. And what happens is that, let's say we've got down here at the bottom, the pink chromosome coming from father, the purple chromosome from the mother. We see that they exchange some of their genetic material. So some of that purple chromosome from the mother is now on the pink chromosome from the dad. Some of that pink chromosome from the dad is now on the purple chromosome from the mom. So now both chromosomes have a little bit of DNA from both parents. Now just imagine that, we've got 20,000 genes. What we are seeing here is crossover of only a singular gene on just one chromosome. We've got 20,000 genes that are spread out across 23 chromosome pairs. So the possibilities are enormous as a result of crossing over. The different combinations you get are enormous. Okay. Here's another image that demonstrates crossing over. You can see that the blue and red chromosomes are going to switch over a little bit of genetic information. All right. Cool. The third source of genetic variation is independent assortment. Independent assortment is a law that states that different genes are inherited independently of one another. In other words, inheriting one gene from a parent does not increase the chances of inheriting another gene from that parent. It's all independent. So if you get your dad's brown hair, that doesn't also mean that you're going to get your dad's curly hair. If you get your mom's height, that doesn't mean that you're going to get your mom's muscle tone. even though these genes might be on the same chromosomes, they're not guaranteed to be passed on to the same gametes. Genes are not linked to one another. So there are so many combinations because they can all be passed on differently. The real world effect of this is that some people have characteristics of both parents. We all have characteristics of both parents. But some people look very much like their father, while they have a sibling who might look very much like their mother. And then another sibling who looks nothing like either parent. Because these combinations of genes and traits are not linked. Any questions about this process or any of the three sources that we've talked about so far? Okay. Two more. Nondisjunction. Nondisjunction can be a difficult one to understand, so I'm going to go through this closely. Nondisjunction is the failure of homologous chromosomes or sister chromatids to fully separate during meiosis. It results in some of the gametes having more or less genetic information than they should. There's not a guarantee that every gamete will be affected. It just depends on when non-disjunction happens, as we'll see on the next slide. But this means that if that gamete that is affected, if an altered gamete is a part of fertilization, then the resulting offspring might have and will have an odd number of chromosomes, as we'll see in a couple of slides. So for now, what I just want you to know is that non-disjunction is the failure of chromosomes or chromatids to fully separate. Literally, junction means to be together. Disjunction means to separate. Non-disjunction means failure to separate. All right. So on the left, in the column on the left, we can see normal meiotic division. So in that first row, the before row, the light blue row, the cell, we're seeing a parent cell that has just recently gone through interphase, so it has just copied its DNA. We've got two sets of chromosomes there, two homologous chromosomes paired up. In this first column, it's normal. So we can see that the homologous chromosomes get pulled apart in meiosis 1. And then the sister chromatids get pulled apart in meiosis 2. And so the end result is that all the gametes are normal. They all have a haploid number of chromosomes, N. That's what you want to see happen. Compare that to the middle column. We start off with the same parent cell. But as we can see in meiosis 1, we've got non-disjunction happening. One homologous pair does not separate, so one daughter cell gets a little bit too much genetic information, and one doesn't get enough, as we can see. This one gets too much. This one doesn't get enough. So all four resulting gametes are altered. 2 of the gametes have too much genetic information. They have N+1. And 2 of the gametes don't have enough genetic information. They have N-1. So all 4 of them have an odd number. On the right side, the last column, non-disjunction, instead of happening in meiosis 1, now it's happening in meiosis 2. So we start off with the same parent cell, the homologous chromosomes split up properly, but now one of these daughter cells, the sister chromatids don't split up properly. That's non-disjunction. So in this case, 2 gametes are altered, but 2 gametes are normal. So one of the gametes has too much, one has too little, but 2 are all good. So non-disjunction that happens later on is less damaging than non-disjunction that happens early in meiosis 1. Okay. What are the real world implications of this? So what we're seeing here is called a karyotype. A karyotype is basically 3D imaging of a person's chromosomes. You can see the 23 chromosome pairs, and they get smaller as you go through from 1 through 22, and then you've got the sex chromosomes. Now, can anybody tell me which sex this person is? We've got 2 X chromosomes. Which sex is that person? Any guesses? That's a good guess, Josue, but it's incorrect. Males would have 1 X chromosome and 1 Y chromosome. Females have 2 X chromosomes. So don't worry, we didn't learn that. But if we look at this karyotype, we're looking at these different pairs. When we get to the 21st pair, we can see that something is off. It's not a pair. In this case, it's a trio. That's not normal. That's not a good thing. This is a result of non-disjunction. This person got too much genetic information from one of their parents. One of the parents' gametes had 2 chromosomes in the 21st position when it should have only had 1. So this person got 2 from their dad and 1 from their, this person got probably 2 from their mom and 1 from their dad for a total of 3. So now they have an odd number of chromosomes. We call this condition specifically trisomy 21, tri, of course, meaning 3, but it's more commonly known as Down syndrome. So I guess I should have included a picture of a woman with Down syndrome. That was an oversight on my part. Now, of course, Down syndrome can result in people having a shorter stature. They're more likely to be overweight. They might have cognitive impairments. And they can also have, they're also more likely to have a shorter lifespan because of heart problems often. But this does not apply to everyone with Down syndrome. And it's important for us to note that these people are differently abled. Some of them do have quite impressive cognitive abilities that allow them to live independently, that allow them to hold jobs that any more traditionally abled person would be able to hold. But people who have Down syndrome have that non-disjunction in their 21st chromosome position. The last source of genetic variation that we'll discuss is fertilization. This is the process by which a sperm and an egg cell unite to create what's called a zygote. It's kind of a fun word to say. Zygote. No, this is not like some futuristic goat or some goat from some other planet. This is just a cell that is the result of a sperm cell penetrating into an egg cell. So this is the very first cell that is on its way to becoming a full-grown organism. After the sperm fertilizes the egg and becomes a zygote, now that zygote is going to undergo mitosis. It's going to start dividing, making copies of itself. And then eventually, some of those cells will go through another process that we'll talk about later in the course called cell differentiation, meaning some of them will go on to become brain cells, some will become olfactory cells that help you smell, some will become taste buds, some will become cells in your eyes, some will become muscle cells. They all become different things, and eventually they start to form different tissues, then different organs. So this process of embryonic development is really, really impressive. It really is a miracle. Okay, so that's what I wanted to go through today. We only have 5 minutes left. There is an exit ticket, so I want you guys to go ahead and knock out that exit ticket, but I didn't include anything new today, no new assignments. So what I want you all to do this weekend is use your time to go over missing assignments. Quite a few of you have missing assignments, even if they are small assignments, like warm-ups and exit tickets.
Do you notice anything seemingly impossible in this
What is
picture?
that?
Wings? Well, yes,
Something's
it's quite
playing.
interesting. But it's not all that simple. This
Oh,
butterfly wasn't
Lance.
just with an unusual coloring. Half of it is male, and the other half is female. How's that even possible?
Oops. Muted you, Lance, but I don't need to pin you. How do I unpin Lance? Unpin. All right. So what I want you all to do this weekend is make sure that you're going over your missing assignments. Go ahead and knock out the exit ticket for Unit 3, Day 4. You should be able to get that done in the next few minutes before a fourth block. If you have any questions or you need anything of me this weekend, please don't hesitate to reach out. We will have a formal assignment that I'm going to introduce. I should be done with it by Saturday, so I'll send you guys a message and you can do that. That'll definitely help you guys who are probably, those of you who are doing well on your assignments, but you're struggling on the tests. This will help bring your grade up. So please do take that seriously, pay attention to it. It'll be worth 20 formal points, so that's a fifth of a test. So that can go a long way, considering we've only taken 2 tests so far. One other thing I was going to mention, I can't remember. So we've only got 3 minutes left. Knock out those warm-ups. I'll be on mute, and then I'll send you off. Okay, it's 1:20. So thank you all for working hard. If you're working on the exit ticket, you should just go ahead and finish it so it's not hanging over you. Other than that, enjoy your weekends. Well-deserved time for relaxation. Let me know if you need anything, and I will talk to you all on Monday.
Have a nice weekend.
Thanks, Lance. See you, Dijon. See you, Melissa.
See you Monday.
See you, JT. See you, Josue. Bye, Abigail.